Today we're going to talk about feral swine, which is one of the big areas of my research and extension program. We've been funded for approximately 3 years from the USDA Veterinary Services and National Wildlife and Research Center to collect information on feral swine or wild hogs in Florida, and it includes lots of projects. So we're looking at population densities, we're looking at diseases they carry, we're looking at what damage they do. And today's talk is going to be about two aspects that we're working on. One of them is rooting, because it's one of the major ways they actually impact natural resources and rangelands. And the other one will be actually diet. And the diet stuff I'm talking about is a genetic study where we made graduate students and myself and a few other people go around and collect fecal samples from the field over a whole year. And we'll talk about some of the preliminary results that have just come back from that. And it's pretty cool to see what they're actually eating. All right, so let's start with drones and how to actually use them. So drones, or unmanned aerial vehicles, get attacked, okay, and cause all sorts of problems for wildlife and people all the time. But we can actually use them to help with research too. And over the last 10 years, drones for the normal backyard researcher have become really easy to get hold of and to use. It's great, you can fly a drone really easy, you can take lots of pictures with them, but what you do with the pictures afterwards is the big issue. And for anybody who wants to get into drone work, it's also the most expensive side. So actually storing imaging and classifying imagery and actually working with imagery is much more expensive than actually the drone itself. So they're used for all sorts of things, and there's a whole variety of them. The two that are mostly used by researchers are quadcopters or fixed wing type drones. We use a quadcopter. They are generally used on smaller areas because they rely on battery power that can't move them that far, whereas actual gliding drones can actually cover a lot more distance. There's backwards and forwards between what you would use, but for our purposes, we've used a quadcopter. And there's lots of things you can do. So you're actually taking a picture of the landscape, and depending on how that picture is taken, you can do all sorts of things with it. It can be a two-dimensional image where you can look at vegetation and habitat monitoring. If you actually take particular types of images very close to each other, you can actually work out 3D imaging as well, which has been around for many, many years. You can then use spectrums of wavelength from those images to actually classify that vegetation, and that's what we're doing. We're particularly interested in damage assessments. So for us to actually do that, the damage you are recording has to look different to what's around it. Okay, if it doesn't, you can't see it. Okay. So some of the things drones have been used at that I think are really cool is things like counting species. So if you were trying to count this tern, for instance, would you want to count it using the picture in the right-hand top corner with a pair of binoculars? 1, 2, 3, 4, 5, 6, 7. Or would you like a picture for, say, B, or the bottom right-hand corner that was taken and you could count it from there? And so what's happening is you're actually changing the way you're looking at a colony, and breeding bird colonies, drones have been used on a lot, and they give a much, much better accuracy of the number of species there. And there's been a comparison study done for that, which is this one. And I can't remember what it was done on. It was frigate birds, I think. So you can see all those frigate birds nesting in clumps of colonies in picture A, and then they asked people to count them from the ground, and then they used a drone to actually assess how many there really were there. And the green is what people on the ground did. The blue is actually the real drone counts. So what you can tell is that people on the ground are underestimating the number of individuals out there to start with, and in general, they're either, not encompassing all the variation that's actually out there, or they're creating a lot more variation. So you can see that in the large green bar and the short bars. could count wood stalks in Florida this way and it'd be a lot easier. So that's true. I mean, so wood stalks are an endangered species. It would actually be a way to actually count the colonies in a much more efficient manner than actually taking a plane up to fly them or trying to count them from the ground. So there are really good applications you could use this for. That's pretty easy type way to use a drone. But our objective is to actually re-fly the same area over and over again and actually take pictures, bring all those pictures together to create a large coverage that you could actually then assess. And that way we can look at changes over time, and we can actually extract the features we want from those pictures over time and assess, for this, in this case, the amount of rooting. So we wanted to do this originally the same place we're doing all of our other work, and we are doing it on 36 wetlands and 24 pasture plots, and you can see that in the map on the right there. So the blue is all the wetlands. Wesley's not here today, is he? So Wesley Anderson is doing work in those, specifically measuring all the amphibian and vertebrate wetland community generally, to actually see if feral swine rooting impacts those communities. And we're also flying them so we can actually show or give an index of how much disturbance or damage they're doing on his wetlands. And he's doing these flights. So I just tell people how to do this. I don't go out there and do all the flights. So we want to quantify that size and the pasture impacts. The top left-hand side, you can actually see typical rooting in pasture that should be occurring right now, actually. They should be increasing their damage on pasture areas and coming out of the wetlands as they dry down. So I tried to do this when I first came here in 2014 with this 3DR X8 quadcopter. Actually, I shouldn't say I tried to do this. I tried to get Bethany to do this. And so I didn't fail, Bethany failed. Okay, that's how it works. Yeah. No, so we actually worked really hard to get this drone to work. And it would work, except that it was glitchy. The actual electronics of the device, the motors, and the way to actually get it to work was really painful. And we kept getting problem after problem after problem. And we did that for about 1.5 years. And we can go through this, but this is how we do it. Bethany did learn how to hack into systems, and so she's got some skill sets that are good for changing how your camera works and those sorts of things. One of the biggest problems we had actually is right down the bottom there was a very short flight time. So it was actually only 4 to 5 minutes at those times, so it was quite heavy. We couldn't really get the areas we were interested in, and changing out batteries in this particular drone was difficult, and it was right at the beginning of when drones started to become easily available. Growth of drone technology became really rapid. So if we'd waited about 1.5 years, we could have got this drone, which we now have, and it's phenomenally better. I mean, you can talk to Bethany over lunch now about how much better this is, but we were instantly able to do what we needed to do, which was a big difference to what was happening before. It's easy to fly. It's autonomous, so you can actually create maps and fly the same route. Once you save that route, you can do it again and again and again and again, which is really helpful. It's pretty simple, and even monkeys can fly. Is that the way I should do it? So we can get, Wes does it no problem at all. All right, so that's been a problem in the past, is actually trying to get people to learn some of this stuff. And when it's really complicated, they make mistakes. And if you crash a drone, that's sort of it for the day or the week or the month, or until we actually find another one. Who's crashed a drone in here? So this particular drone has software which allows you to just have an iPad, and you can actually almost draw where you want to fly. It's actually quite impressive what they did. You can see from the iPad what the drone's actually seeing. So there's communication backwards and forwards while the flight's happening, and it's got a much longer flying time, so we can actually cover a much bigger area, which we needed to do. And when I say big area, what do we think it's about? 10 acres easily in 20 minutes, that is. Yeah. And in 20 minutes, but what we're able to do is land it, swap batteries out, and the drone's smart enough to remember where to go back to and continue its flight. So you can actually continue to just keep mapping and mapping and mapping. So we've actually flown all of the woods here at Ona as part of what we're doing as a baseline of vegetation, and we've been flying the pastures and wetlands at the ranch for almost 2 years now, and so we have quite a lot of data coming up. So all of that, you get imagery, and imagery comes out, and you get hundreds of images. And so what do you do with them? Do we sit there with our little bits of tape like I used to do, and you stick them all together and we create a really big board or a mosaic, and you say, great, this is good, and then take another picture of that and say, that's how I'm going to analyze it? Of course not, but that's basically what we're doing, is that all those images are geo-referenced, and so they overlap with each other, and the software that's been created, not by us, but by a number of drone companies now, is so smart that if you've got a 70% to 80% overlap in your image, it can align them perfectly. So it actually looks for variation in the pixel colors and pixel arrangement and does it mathematically. And so you get a big mosaic that's created. We also geo-reference when we're flying using ground control points, because as you all know, your GPS is not accurate to exactly where you are. So it's going to be accurate to potentially 3 to 15 meters, depending on how many satellites you have. So we have to take that into account because of the work we're actually doing. So we've got a pipeline, and then you can see this picture in the bottom right-hand corner, and it's a wetland. The green boundary is the supposed boundary of that wetland that was mapped. You can see a truck in the top right-hand corner of that, well, left-hand corner of that picture, and below the truck you can see a whole series of white openness. That's pig rooting. Okay, that particular pig rooting is outside of the wetland, but inside the wetland you can see everything that's been identified as red, and that's been outlined, and that's actually rooting within the wetland. And that process is a separate process that we actually do in GIS, and that's Geographical Information Systems, and I'll talk about that a little bit in a minute. So when we actually use these drones, this is what we see. So this is straight off the iPad, and the reason I'm showing people this is a lot of people haven't seen this sort of stuff, and it's actually a lot easier now and can be implemented by lots of people to do this sort of research. So you can open it up. We can actually set a whole lot of parameters, how high you want to fly, how much overlap you want, and decide what is our ground resolution. What is a pixel in our picture actually going to be? So in this case, we've set it so that the resolution of our pictures is we're actually getting reflectance value from a 2 centimeter area. So when we mosaic all that together, I can tell what the reflectance is of a 2 centimeter area. That's important because once you build all those 2 centimeters together, you'll be able to tell whether it's a grass clump or whether it's bare soil. And you can get right down to almost species identification if we had enough variation in the species reflectance. We can adjust flight paths, and we can tell it to do the best things like missing, you know, overhead power lines, the local bald eagle's nest, which you saw at the beginning. So got to take those things into account too. So when we did this is what ended up happening. At 2 to 2.5 centimeters resolution, our smallest wetland took us 35 images to cover. Our largest 40-acre pasture is about 400 images. So they all come down for one coverage, and then they all get stitched or mosaicked together. And so these are single images you see here, and then eventually they will be mosaicked like this. And we do this online. We don't want to do it here. The actual software to buy this to actually do this is thousands of dollars. And right now it's better for us just to actually send it, and we get it back from a mosaic, and we upload that. So you know when the internet's really, really slow here? It's not us at all. It's never us. No. It's not actually. We only do it at night when all the students are watching movies and we're swearing away, going, God, can one of them turn that movie off for at least a minute so we can get the rest of these images up? So we download this mosaic, and in general you can see the dark blue areas on there. There's actually, that area means there's 13 images that have actually overlapped at least in some proportion there, and it's all together. Because if you remember, I said we usually overlap between 70 and 80%. So each image is just a little bit overlapped, and that's why it takes so many to get to this point. But when it is overlapped, this is what you get. If I zoomed in on that, you'd be able to see a clump of andropogon or a patch of spartina grass, the top of a palm tree, really easy. So that top one there is about, 135 images, and then this pasture down the bottom is 430 images. So we have this flown for all of our 24 pastures and all of our 36 wetlands, and they're repeated over time. And this is that same wetland with the truck and the routing, and you can see, we can zoom in and see a lot of this really easily. The last thing, this is really for a research perspective. If you want to get really accurate, you make Wesley go out and put crosses all around the wetlands and all around the pastures that stay in permanently, and this is a known ground control point that we have a very, very accurate GPS on. And so we're within less than a meter to knowing where they are, and then we can geo-reference all of our imagery back to that. The reason we did that is that we know that we can be off a little bit, and so when we're talking about area, whether this matters or not for the project of interest, you can be up to, or on average, about 3% error in your stuff. All right, so we have all our images and we have a big mosaic, and we pull that mosaic into ArcInfo, a geographical information system, and then we do what's known as an image classification, which is really just a funny word or a explanation of what are the color bands that are actually in that pixel. And this particular camera takes, like most cameras, has 3 color bands in it. Do people know what they normally are? So when you take a picture, all these digital cameras actually have three color bands, and it's a layer of 3 colors that basically defines what you're seeing. So we're separating them out, and it's hard for me to show you, but so you can see band 2, band 3. Actually, band 3 is up there twice. I don't know why that's true. Oh, it's band 1 compared to band 2, band 1 compared to band 3, and then band 2 to band 3. So what we're seeing here in the red and the graphs is pig rooting, okay? And if you compare band 1 to band 2, you can see those red pixels completely overlap the black pixels, which is non-rooting, okay? Does that make sense? And those scatter plots have been defined by these polygons drawn in the image. So those big polygons you see drawn there, we just drew them with a cursor on the computer, and we said this is non-rooting, and it took all of the different color parameters out of that and said all of these colors represent non-rooting, and it does it for 3 bands. If you're looking at band 1 and band 2, you can't separate them because they're completely overlapping. All right, so that particular color, and maybe it's green, maybe it's gray, white, whatever it might be, in terms of the color reflectance in that band, it's not defining rooting and vegetation of the landscape part. But the others are. So band 1 and band 3, they start to separate those really well. And so we take that and you then use that as a model to reapply to the whole image. So we're just taking a little snapshot of it and we reapply it. And this is what we get. And so we get all the red is potential rooting, and you can see it's spread all over the place, some tiny, tiny little bits and some much larger. We then step through a process where we say anything under 10 centimeters we don't care about, because that type of rooting, we don't really know if it's truly rooting or something's just dug something up, dirt. Then we have a few other things, is that if it's less than 30 centimeters apart, of larger than less than 10 centimeters, they all get aggregated together. And so the minimum area of a plot has to be 1 meter square. And if it's a really big area, you're not allowed to have holes in it that are greater than 400 centimeters square. There's just some rules. We create rules, otherwise it gets too complicated and you'll just get a mess. So we apply all of those rules, and it goes from this down to that final product, that aggregated polygon. And this is almost automated, okay? So it does all that. We get from really messy, and we went all the way down, and then we throw it back up, and this is where you still need humans, okay? So robots can't do everything. Wesley's definitely not a robot. He can't perform the same task consistently over and over time the same way. It always changes depending on whether he was out that weekend or not. So, but anyway, he what he's been doing is he puts these, I think you can see the yellow grid over the top there. He'll put that yellow grid over, and he'll step through that grid, and he'll just say, okay, does this grid look proper to human eyes as rooting or not? And he zooms right in to that grid, and he does it to a 1 to 150 map scale. And he can tell, if you look at that incorrectly classified bunch on the right-hand bottom corner, that's Spartina grass. Can you see how its pattern is really weird? Sort of spread out, it's like just clumping like this. It just happens that it's all dried out and it's giving a really similar reflectance to rooting, which is on the correctly classified polygon. So at that point in time, he says, okay, we're going to remove these clumps that are Spartina. Now, if Spartina and rooting were right next to each other, which does occur, this would be actually a problem, and we probably get it wrong, and that happens. So this is only still an estimate, it's not 100% truth. Anyway, so he does all of that, and then this is what you ended up with, which is what I showed you at the very beginning. And in this case, 0.7 acres of this wetland was rooted, and that means about 2.3% of the wetland was rooted in that particular time frame. And we do that. That's been done for everything. Over time, this is what you can build, and we can see that in this particular wetland from November 2016 through to March 2017, the locations of the rooting were the same, and they didn't change that much at all. Okay, so it was probably rooted once and never grew back, or they're just returning to the same spot, rooting the same patch all the time. It's important to know. On this pasture, in this particular case, rooting's increasing over time, so they're actually come to spots, and then they're returning to spots, and those spots are actually growing in size, especially along the ditch. So you can see on this far ditch one. So rooting causes, from other studies we've done in pasture, and you know this from other talks I've done here, 44% forage loss in improved pasture. Okay, so if it roots an area, it loses 44% of its capacity to produce forageable grass for that year. Okay, and what that means is about, if 2% of an acre is rooted, these are old figures, but it's approximately a couple of dollars is lost towards calf production. Another way to look at this is if you're on a 5,000 acre property, you'd lose 100 acres of palatable grassland to rooting if it was 2%. So those of you out there, how much can you produce off 100 acres? How many calves could you produce? Yeah, calves. 50. Damn, I'm coming to your place. So 50 calves off 100 acres. So you're losing that product on a 5,000 acre ranch. If 2% of it's rooted, that's what you're losing. The ability to produce 50 calves. Does that make sense? And whatever those calves happen to be worth in today's market, these particular prices were when the calf prices were high. So you wouldn't lose as much. So is it worth our while to let the rooting continue on these pastures when you think about it that way? That's the choice of the landowner. 2% rooting is probably low for quite a few of these places, and it's a lot higher. All right. How much does it cost? A little bit, but I think it's relatively cheap for a research project. If you wanted to do it yourself, I might not recommend it, but we probably did all this work and we're continuing to use this equipment, and I know you could look at this, it was probably about $5,000. And it's going to be used for a lot of other things. So we can use it for vegetation analysis, gap analysis, feral swine work still going on. Pretty pictures for Andrea, videos of the place, so we can actually take those too. So if anybody wants themselves riding along a tractor, aerially done, you've got to talk to Bethany. So you can put it on your poster. So this is what we're using it for. You've seen rooting damages, temporal rooting damages. We're really interested in how it impacts amphibian communities because that's unknown. There's been one study, maybe two studies now, ever done. One of them was in North Australia, and then the other one, sort of indirect. And that was actually done in the Panhandle of Florida. The other thing we want to do is once you actually do all of this damage assessment, and if you come in and you do a removal effort or a control method on a population of swine, and you're not quite sure how much you remove, hopefully we know a little bit, how much does that actual damage drop? And so that means how much effort you're putting into cost of control of swine, what is it returning you in terms of less damage? And that trade-off cost is an important thing to actually look at. All right, so we do all of this rooting stuff, and I've got about 6 more slides in here because this is all preliminary. What we want to know, what I've always been interested in, and people always ask me this question, is what are hogs eating when they're performing all of this rooting? Jeff, what are they eating? Grubs and bugs. Okay, I agree. Grubs, bugs. Oh, you forgot one. Tubers and tubers. So plants, invertebrates, and other bugs, grubs. And he's right. That's true. They're eating grubs and bugs. But sometimes we want to know exactly which ones they are and what the breadth of that is. So in this study, which is really a study on the genetics of feces, so we sent people around to actually collect fecal matter from the field. And the objectives are, what do they eat? Collect fecal material over a full year so that we actually know how their diet changes over the year. Be able to extract DNA from that feces, which is not that easy in some cases. And then the way you do it is we're actually using sequencing. So everybody knows what their DNA looks like, don't they? I'm only teasing. We're going to have a lesson in a minute, all right? And there's a test afterwards. But what we do is we take a little bit of that DNA, that sequence DNA, and we call them barcoding genes to determine the species composition. So it's a little segment of DNA, and we actually compare it to known sequences to actually understand what species is in this feces. That's great and good, but like all machinery and computers and all these great tools we create, they're often wrong. And if there's something similar in there, like a ranid frog from Southeast Asia is in the database, but the local ranid frog here is not, who do you get coming up on your sequence as the most likely species to match? The guy in Asia. And so if you actually don't use some other filtering tools like local experts' opinions of whether or not that species actually exists in the area, you're going to be really wrong. And this happens a lot in these types of studies. And so we're at the stage where we're using local experts to actually go filter through all of this data to actually decide what the species that was returned from the known databases actually is. And I'll show you some of that. And the last thing is, how does the diet change? So I call this the dispersed sampling method. It didn't start off like this. It started off very, very methodically. Let's do it methodical. Let's do a transect. We're going to walk this transect for a mile. We're going to pick up all the fecal material along that transect. If it's fresh, you can keep it. It needs to be fresh. Old fecal and old DNA is not good. So anything that's bad, you throw away. All right, and then you rerun that transect the next day, because you know then any fecal material deposited is less than 24 hours old. Sounds good, doesn't it? So what do you think happened? Go get everybody walk a mile, 10, 20 miles is walked, fecal samples, old fecal samples are thrown away, and they went back and re-walked those 20 miles. What did they find? Nothing. So pigs were not coming back to those sites and depositing on a regular enough basis to make it logistically worthwhile. So the dispersed sampling method is when people randomly walk around and try and find a pig toilet. All right, so they're just going to move around, and the best way we can actually account for repeated going to the same place is we make sure that they do it across a dispersed pattern. So you can see there's a number 1, a number 2, a number 3, and a number 4, and a number 5 there. They were defined sectors for each part of the ranch, and there had to be 5 fecal samples collected from each one of those sectors, and not 25 from one single location. So you spread the collection out. The red dots you see up there are the final dispersed method collections. They're not random at all. They're definitely clumped, and you know pigs are going to certain areas, and that's why. So we did this, and we did it 6 times a year, and each time we collected at least 25 fresh samples, which is a minimum of 150 samples, and in the long run we actually ended up collecting 204. So 204 pig poop samples. And they are then going to go through a process known as next generation sequencing, which is basically doing it really fast. That's all. It's a fancy word for doing things fast. All right. You have to extract DNA, and then what I was telling you before is we actually compare that to what we know as DNA barcodes. The idea of barcode is that it provides a rapid way to accurately and automatically identify a species against a database. And it uses a specific part of a DNA sequence, and it's called an identity tag. This is my writing here. By default, a good barcode will vary across the species of interest. Why would I say that? This is your first question. Why does the barcode have to vary among species? In order for you to read it. Read it in terms of, what do you mean by that? So you'll read a sequence from a species. Okay, so you've got this sequence of DNA, you read that sequence, you know what that sequence is, and you're comparing it to a known barcode. All right. So why am I saying that barcode has to vary? That's the question. Nobody going to answer? So maybe I'm asking the question badly. So the barcode has to vary because if it doesn't vary at all, every species would be the same. You'd get everything. So if that's the case, that's known as conserved DNA. It doesn't change at all. And so there's lots of places in our DNA where it's conserved. You get the same sequence all the time. Your sequence would be identical to mine. Yours would be identical to mine. And actually yours would be identical to a gorilla and a chimpanzee, and that's actually true. All right? And even in some sequences to a snail or an amoeba, our sequences are identical for parts of that DNA. You can't use that part of the DNA because we're all the same. So a good barcode has to be highly variable, all right? And it's variable and specific to a species. And the bit in brackets is this is not always the case. It's actually quite true. So sometimes it's really good at defining some species, and sometimes it's not. Anyway, I wanted you to know that because it's really important if you ever read stuff about genetics. So we use 3 barcoding genes really for that purpose. So we use one that's known as tRNL for plants, and we use one for CO1 for most animals, and then we use one for 12S because it's better for amphibians, okay? Because the CO1 doesn't do very well on identifying amphibians. When we do that, we identify this sequence and we look at percent similarity, and this is how it's done. You didn't know you were coming to a lesson, did you? All right. So this is percent similarity. So when everybody's talking about these next generation sequences, these great big genetic studies, and they go, oh good, we're fantastic, we're doing all this work in DNA, it's really they're comparing 2 things. Actually, well, it's 4 things, but they're really only comparing 2 things. They're comparing whether or not A in this line is in the same position as A in this line. Okay, that's it. That's how simple similarity in DNA sequencing for barcoding is. A and A. I'm coming back to the screen. So if you look at this particular sequence, this is our barcode gene we want to look at. It's a bad one because it's really short. It's only 10 base pairs of the DNA here. Where does it vary? So they're the ones in bold. We've got a T and an A, on the second line, then we have a G and a C and a C and a T. So in this case, our sequence is the top one, all right? We tried to identify it against a big database known as BLAST. It's the biggest one in the world. And it came back telling us our best match was this sequence that it returned. Now the similarity of that match is 70%. 7 of those 10 pairs matched. Okay. Would you accept that that was the same species at 70% similarity? How similar from the known DNA sequences genome of humans are they to chimpanzees? Do you know? Look at that. So if you're 70% or if you're 30% different, you're actually nowhere near 98% similarity. How far away could you be in terms of identifying this species accurately? A long way, all right? So you throw all these things out. And we generally don't use anything unless it's above a 98% match, okay? It has to be a 98% match. Hopefully higher if you've got something good in the database. So we did this. It's a much longer sequence of DNA, some couple of hundred, couple of thousand of these pairs, and we do this sort of thing. And we could create graphs like this. Hopefully you can see it. But what it's saying is this is for tRNL gene, and these are plants along the bottom. They're actually families of plants. So I haven't even gone to species here because that needs experts in plants to do it. But the families are probably right. We collected 204 samples, okay, fecal samples. And what this unlabeled y-axis here, that's really bad. Don't ever do that, okay? Students out here, don't ever do that. But this is actually the number of times that family was picked up in those 204 samples. And so it's a yes or no function. Yes, it had this particular family of plants in that fecal sample, or no, all right? The first one you see, this is a question for Brent, is 191 of 204 family Poaceae, it's a really easy question, were found in feral swine diets. Do you think that's a reasonable thing to say? And why? Yeah, so what's Poaceae for everybody here? So Poaceae is grasses, all right? And so it's saying the family of grasses, 191 of the fecal samples we collected had that in their diet. It doesn't say which grass yet, but 191. So that makes a lot of sense. Okay, and basically you can see that this is a pretty diverse lineage of families. But what I can tell you is there's 278 different plant species that we identified so far in 204 fecal samples. Okay, I think some of them will aggregate together because we're going to have the same problem of this similarity where particular species were being chosen over others, which is not correct. I'm not going to delve into it because this is just preliminary, but I thought you'd be interested in this. And then this is just a graph of the same families just over time. I was quite surprised with this because if you actually look through each one of these graphs, in general they look very similar, except for some of the, I'll just say the orange colors, not the reds, the oranges are actually changing quite a lot over time. It's a little hard to see this because there's just so much data here. But this is basically time points of 1, 2, 3, 4, 5, and 6. The 0 up the top is samples that were not time collected, had no timestamp on them. But diets are pretty similar over time, which I'm surprised at, and for the plants, and I think we want to delve deeper into this. So there are other things. So that's just a plant gene. This is what's known as the CO1 gene for animals. And here's a good question for somebody. Who wants to have the next question? Hands up. Who knows lots about animals? Austin. Huh? Austin? This is families, so it's hard to know, but does anybody know what the Muridae family is? They're little, they got whiskers, got a little tail. Yeah, so moles, groups, mice. So this family here, Muridae family, the 60 of those 204 feral pigs had rats or mice in their diet. And they're probably eating neonates or from the nests in the grasslands, and they can find those. I was pretty shocked at that, but then I started looking at literature, and yeah, it's pretty common. So there's some other things in here. Oh, you guys should all know this one. So bovidae. Bovidae. What's a bovidae? That's a cow. Come on. Yeah, good. It's a cow. So if you look at this, there's 48 cow occurrences in these fecal samples. Now, you ask that and you say, okay, 48 cow occurring in a diet. Does that mean they're eating cows? They might be eating dead cows. What else could they be eating? After birth. What else? Feces. Placenta. Yeah, but they could. One of the things that'd be interesting, if they are eating placentas, we should see it in the time series. So you should be able to pick that out. If they're just eating feces, for instance, cow feces is probably pretty nutritious to a pig, especially if we've been feeding them lots of molasses and all sorts of other things. It comes out the other end. I think they'll like that. And
you see much cannibalism?
Not here. And we can't tell because remember these all came out of a pig, so they all have pig DNA in them. So all of them are eating themselves. but it's a good question. So nobody's asked me yet, but this one here, this one sort of makes sense too. This is, I can't even say that. I'll try my best. So it's Glossa scholicidae. This is a family. 163 of those 204 samples had it in it. And you almost said it before, it's actually worms. So that is worms. And so this is really interesting to me because we're identifying that most of these pigs have actually got worms in their diet. So that's probably a large component of their diet that they're targeting. And there's all sorts of things we can go through along here, but there's some of the interesting ones. Jeff will know what cervidae are. That's deer. So 22 of these pigs had deer in their diet, in these fecal diets. What's another good one? All the way down somewhere, there was one that was really interesting. It came up as alligator, and that was a great one because I don't know how this happened, but it wasn't me. I guarantee it wasn't me. But somebody actually found an alligator fecal material and decided to call it pig and put it in the bag, and we sent it. And we can tell that because what ends up happening is the organism that actually produced the fecal material dominates the DNA that you're getting out of that fecal material. So we have ways to look at that. And if it's astronomically huge, then that's part of the problem. Anyway, so there's some diet things, and this is just the same sort of graph. And we can talk about this over lunch as well. But what I'm telling you, which you probably already knew, is that feral swine diets are really broad, and they use a lot of resources to survive. And a couple of the other things we've looked at is they're targeting oak a lot, and acorns look like they're in their diet the whole year. So they're actually finding probably a lot of fallen acorns all the time, or chewing on roots as well. And the propensity of acorns to be in pig's diet is more starting in November, December, January, February, March time periods. And I'm going to leave it at that. And you got any questions? Yeah.
So you're showing just families. Are you planning to go and look at species?
Yeah.
And then like tell what's going down?
Yeah. So I'm more confident that families are correct with the similarity idea. But when we get down to species, the exact thing that I was telling you was happening to us. So that when we actually compare it to a known database, we're getting frog species out of Asia, we're getting worm species out of South Africa, and we're getting species that happen to be in the database, and their DNA sequence is pretty similar to what we're showing, and the local species that they're eating is not in the database. So we actually have to use local knowledge of similarities to actually look at that. There's quite a few examples of that in the plants too, and so it's one of those things that you've got to. It's logic. You've just got to be smart about how you look at your data, especially this DNA sequence stuff. It is the first study that's ever looked at the sort of temporal diet of pigs across the year, and it's a pretty good idea of what they're going to be eating in Florida. I've learned a lot just by looking at the data for a couple of days. It's pretty amazing. Any other questions? Nope. Well, thank you everybody for attending. Thank you for those online. If you want, you can send me an email. It's rboughton@ufl.edu. Thank you.